Amazing. Thanks, Andrea. Never seen this before. QR code for acknowledgement. We don't do questions now as in the previous session. Please go to slido.com, HCA COVID-19, and put your questions in, upvote others, start discussion there, and we'll have a Q&A at the end of the thing. Let's move to our second speaker in the session. We're happy to have Roland Eils here presenting about his recent work on crosstalk between airway epithelium, also on the single cell level. Roland is well known as an active member in the Human Cell Atlas, in the board, but for me, sort of more personally, has been one of the godfathers, I would say, of systems biology in Germany, driving so many things. And so when I started as a junior group leader, there are a lot of activities that I'm really grateful and happy that I was able to basically do due to many of Roland's activities. He's been active for a long time at the German Cancer Research Center in Heidelberg, and then moved to found as a founding director at the Berlin Institute of Health, the Digital Health Center, where he's now translating a lot of his sort of more general work before into clinics. I think that's where one of those quite fascinating projects stem from. So we're looking forward to your presentation, Roland.
Yeah, thank you, Fabian, for the very nice introduction. I'm switching gears a little bit in my presentation compared to the earlier presentation, in particular in the first part of the session, because we are looking at the crosstalk between airway, epithelium, activated immune cells, which we believe defines severity in COVID-19. So when the pandemic, before the pandemic arrived in Germany in sometime around February and March, before we even had the first COVID-19 patients available for our studies, we were very keen to look into baseline information now about the airway, epithelial, and immune cells, which might be most susceptible for SARS-CoV-2 infection. And we were very fortunate that in a very completely unrelated study, we had access to lung samples from healthy donors, and where we could study both the cell type distribution and the propensity with respect to ACE2 expression and its cofactors in different sections of the airways, or from the upper to the lower airways. And in a nutshell, we showed that secretory cells in the bronchia and AG2 cells in the lung are, according to ACE2 expression and its cofactors, most susceptible to infection. And as Aviv already mentioned in the discussion, there are a couple of other studies having looked into similar questions, and there's a meta-analysis now having been performed under the guidance of the Human Cell Atlas of the lung. Now, so then in March, when the first patients arrived at the Charité University Medical Center, we set up a clinical cohort study, a prospective clinical cohort study, integrating all of the COVID-19 patients that are hospitalized and consented to be participating in the study at Charité. And so we did the straightforward thing. So we performed 10x based single cell RNA sequencing from different parts of the airways of our hospitalized COVID-19 patients, starting off with the nasopharyngeal area, so the upper airways, and then going down to the bronchia and the deeper lung. Now, the first study group which we looked at were composed of critical and moderate cases, where we were looking at 2 severity groups and 5 additional controls. And for those 24 individuals which were part of this study, we obtained 36 samples. So obviously we did longitudinal samples along the disease course of those COVID-19 patients to understand also the differences and similarities in the development of the single cell contribution of those individuals. And as I already mentioned, we were looking into 3 anatomical locations from the nasopharyngeal swabs and then further down to the bronchia. And when you're doing these single cell RNA sequencing studies, of course, the first step is to identify all the different cell types which you're looking at. And it's quite interesting if you then look at the change of distribution from in the epithelial cells, but also in the immune cells down here, And if you walk from the control through the moderates and the criticals, you can identify individual cell types like the basal cells, which are diminishing or depleted while you increase the severity of disease, and for example, also resident macrophages are being depleted with increasing severity of disease. And of course, there are other cell types which are increased with increasing severity of disease. For example, on the epithelial side, the secretory differentiating cells, those are the most susceptible to SARS-CoV-2 infection, actually massively increase with severity of the disease. And of course, on the immune side, you see a massive explosion of neutrophils from 0% and controls over 27% in the moderates and up to 61% in the critical cases. Now we then we're turning around to understand the interaction between immune cells and epithelial cells. And quite interesting, we were motivated by this very simple observation shown on the left-hand side, that the interactions which we observed between cytotoxic T cells, for example, in this example, and the percentage of ACE2 positive cells plotted here for ciliated and secretory cells, so two very prominent epithelial cells are involved in the pathophysiology of COVID-19, we observe that we have a very strong association between the ACE2 level, expression level in those cells, and the interaction counts with cytotoxic T cells. And interesting, if you look, for example, into interferon gamma, it's strongly expressed in cytotoxic T cells almost exclusively along the different cell types. And accordingly, in the target epithelial cells, you see an upregulation of the receptors and further downstream regulators like the transcription factor STAT1. Now, if you do this more systematically and look at the interaction of all epithelial cell types with all immune cell types and across the epithelial and the immune cells, and you put up this in the matrix, the color actually encodes the strength of interaction, you see remarkable differences. So for example, for cytotoxic T cells, which I explained in the previous slide, you see that you have moderate interactions with both immune and epithelial cells in the moderate cases. However, in the critical cases, you see a massive increase of interactions between cytotoxic T lymphocytes and epithelial and immune cells alike. Likewise, another cell type, which is has become a very prominent immune cell type in the COVID-19 pathophysiology, are non-resident macrophages. And again, we see the same picture, that we see a massive increase of interaction while you move up in the severity of disease. Now, we then try to understand what are the major factors in those immune cells, the cytotoxic T cells and the different macrophage subpopulations, which actually drive the interaction with the epithelial cells. So for example, for cytotoxic T cells, while you're moving up the severity of disease from control moderate to critical cases, you see a massive increase both in expression as encoded by the color, but also the number of cells as encoded by the diameter of the circle, of a number of cytotoxic mediators, but also the receptors of cytotoxic T cells. Likewise, you see the same kind of observation for monocyte-derived macrophages, but also non-resident macrophages, where you see a massive increase both in expression and also number of cells, both for a couple of chemokines and cytokines. Now, just to remind you, we were looking into, this is all data from the upper airways from nasopharyngeal swabs. But however, as we know, the pathophysiology, in particular severe and critical pathophysiology, develops further down in the airways, in the lungs, in the bronchia. And for this reason, we actually took from the exactly the same patients, so for a subset of those patients, we took samples from the nasopharyngeal swabs from the upper and the lower bronchia. And as you can see that all the factors which we identified to be critically involved in the interaction with between the immune and epithelial cells, they are all massively further upregulated both in numbers of cells and strength of expression while you're moving from the nasal to the upper and lower bronchia. So this actually led us to answer it, pose a number of clinical questions. And just to make a long story short, I would just like to emphasize that there is a potential impact for the treatment of those patients, in particular the critical COVID-19 patients, because what we identified is a critical axis of communication between the chemokines CCL2 and CCL3, which are massively expressed mostly in the monocyte-derived macrophages and non-resident macrophage, and they do interact with a number of receptors, CCL1, 2, and 5, which are then expressed on the epithelial and also the immune cell side. And it has been shown that the binding of those ligands to chemokine ligands to its receptor can facilitate macrophage recruitment, and that blocking this axis may alleviate immune hyperactivation in severe cases. Now, interesting, blocking CCR2, the interaction between CCR2 and its ligands is being used in HIV treatment. But since we don't see an awful lot of expression of CCR2, nor in the epithelial or in the immune cells, we believe that the CCR1, CCL2, and 3 axis might be in a more promising axis to inhibit in critical COVID-19. And actually, there are antagonists available which are blocking very potently the interaction, for example, between CCR1 and CCR3. So this has been a very fruitful and intense collaboration between a number of groups on the clinical side at the Charité and the genetics and epigenetics group at the BIH. I would like to highlight in particular Leif Sander and his team here on the clinical side, and Irina Lehmann and Christian Konrad on the BIH side. And part of our patients were also coming from the University Hospital or University Medical Center at Leipzig, where I would like to acknowledge Sven Laudi. Now with this, I would like to finish my presentation. I'm happy later on to discuss with you.